Culturing viruses in the lab. Remember Robert Koch? That was one of Koch's postulates, that you need to culture it in a lab. Well, if you're dealing with bacteria, you could put it on agar. You can give it its nutrients, things that it needed to survive, to culture it in a lab. How do we culture viruses in the lab? We have to give it what it needs, right? What does a virus need to replicate? It needs a host, okay? So we need to give it a host, a cell that it can invade, live in. It needs a host. All right, so there's a couple different ways that we have done this in the lab. If it is a bacteriophage, and I'll go back and forth to the pictures. If it is a bacteriophage, a virus that infects bacteria, well, that's easy. We grow the bacteria in the lab, and then we infect it with the bacteriophage. What you see happening is you do a lawn growth, so we're spreading bacteria all over the cell, letting the bacteria grow on the cell, and then we're adding the bacteriophage to these healthy cell cultures. What we see, instead of colonies, you kind of see the reverse. You see whenever a bacteriophage lands, it's going to invade that bacteria and kill it. And so you see a hole, an area where the bacteria is not growing. This is defined as a viral plaque. So it's the absence of bacteria growth. And why? Because there was a bacteriophage there that got into that cell and lysed it and killed it. And so now that bacteria is not growing anymore. So this will stay. The virus is going to replicate and move through it. And then you're going to have to keep inoculating a new plate. So this bacteria, we can do plates with that. All right, but what if it's not bacteria? What if it's a human virus? Let me go back to the next slide. If it is a human virus, well, or an animal virus, one way that we can do this is embryonated chicken eggs. Chicken eggs is a large, they're sterile, and they have a lot of different cell tissues. So again, I keep going back to me. You don't want to see me. A lot of different cell tissues here. So if I look at that, I've got embryonic cell tissue. I've got different types of cells within that. I've got different things on the amnion. I've got all of these other things that I don't even know about. There's lots of different places that could serve as hosts for different types of viruses. Again, viruses are very specific on their host, but this chicken egg allows us to put these viruses in, and they can live there, and then, we can just keep them going, and do that. So we can grow viruses in chicken eggs from that. This is the reason when you go to get some of your vaccinations, you may have had the question, are you allergic to eggs? And it's not because that virus has anything to do with eggs. It's because that virus was cultured in an egg, and it's possible a protein could get swept up into the virus, into the vaccine that you're having. So that's the reason they ask you if you are allergic to eggs when you get certain vaccines, because we culture them this way. Okay, so that's one way. Another way we can culture animal viruses, providing them a place to grow. And this is a great sterile, easy way to grow stuff. What about those ones, like, remember hepatitis only infects human liver cells? So to give it a host, I need a human liver. They don't let you experiment on people like that anymore. What we have done is we took human liver tumors. And so if you remember, that tumor is a growth of cells, of host cells that shouldn't be growing, and they've lost their ability to stop growing as they continue replicating. This is cancer. This is a tumor. So say you have a person that has a liver tumor. We're going to take out the liver. It's a surgical candidate. That's not take out the liver. Take out the tumor from the liver. You can take that tumor and keep it in a cell in a laboratory. And because it will continue to grow, if you give it the right nutrients, it will continue to replicate. And you can establish a cell line made up of human liver cells. Now, they're not exactly right because those proto-oncogenes, there's some mutations, but we can keep it in a lab. So for a disease like hepatitis, we can provide a human liver cell and a cell line that'll keep replicating that we can use to culture those viruses. Cell lines was a really big deal when we came up with those. It was in the 1950s. There's a book. I write this down. I know I'm talking a lot, but I love this book. It's called The Immortal Life of Henrietta Lacks. And it talks about the original cell line culture. It's a cell line called HeLa cells. And at that period in time, they were naming cell lines based on the first two letters and the first two letters of the first and last name. So this was the HeLa cell line. It was the first time they got cells to grow in vitro and continue to grow. It was from a tumor that they took from this woman. Lots of great interest in this. It reads like a novel. It goes back and forth. Her family was poor. They were Black at a public hospital. They weren't very educated. The doctors didn't bother to explain what was happening. They created this cell line. The family didn't know, and it made a big deal. It furthered research in diabetes and a lot of diseases that we weren't able to really study because we didn't have a human host. So this was huge. But then later on, when DNA came into play in the 80s and 90s, they contacted her descendants because they wanted some DNA samples to go back and trace this, and they were related. Again, they did not explain what they were doing, and these poor people thought that their mother was still alive in a lab. Oh, it was horrible. Just the ethics involved and the racial undertones. It was a horrible thing that happened. But we learned, and nowadays you have to sign forms for everything. Can you use my cells to do this? Can you do research on this? You have lots of different types of consent forms that have to be signed, and it's due to unfortunate errors in judgment as people were working through and not understanding how this went. So anyway, it's the author is Rebecca Skloot. And after you finish this class, because it's summer and you have no time to read, that's a really good book if you're interested.